We have these brief bios of everybody who's talking today and tomorrow, right? And I was looking at their bios and I was thinking, well, I was thinking about my own career as it dies away. And I realized that for me and many other people, you start off fascinated with some scientific subject or other. And actually, as you go through your scientific career, you keep coming back to it. So in my case, I started off interested in something, how T cells recognize antigen, in 1969. And I'm still, in 2013, studying the same bloody thing. So how can that be? Well, there's more tools and all that stuff, but whatever. Anyway, I was looking at the careers of the people who've talked already and are going to talk in this session, and I was thinking, how can that be true of these people? They're all over the place. Even the first speaker, I have no idea where the hell he's going. Or where you... Yes, well... Well, that too. Anyway, so today we have Matt Todd, and for example, he's geographically all over the place as well, right? He started off in England, Manchester. And he saw the light and moved to Australia, Sydney. Good for you. And today he's going to talk about drug discovery, but how it all fits together, you're going to tell us. Thank you.
So thank you, Larry, for the invitation. It's really great to come to meetings where there's an actual variety of people with personality as well. It's a real pleasure. You know, I was thinking about the Rube Goldberg thing because I'm originally from England, and Rube Goldberg in England is Heath Robinson. So it's the same kind of idea of these drawings of simple things being achieved with elaborate means. And I guess the thing I'm going to talk about today is a little bit like that, because the simple thing is working together, right? That's easy, isn't it? But the elaborate means that are involved, some of it looks quite technical and tricky, but it's the means that we've sort of invented in these two projects to help people work together with no barriers. So it's actually quite apt. So there are two stories, and then some analogies at the end. So the first story is about neglected tropical diseases. So this is a project that I got involved with a few years ago. Praziquantel is, if you ever want an example of a wonder drug, praziquantel is a great example. It's currently given to 100 million people. The numbers are really extraordinary with some of these neglected tropical diseases. Neglected means that the amount of suffering is large compared to the dollar input in research. So this treats a very unpleasant disease called schistosomiasis, or bilharzia, which is mainly in sub-Saharan Africa, and is one of these really awful diseases because, for two reasons. One is that the infectious agent is highly optimized. It's like version 5.8, right? This thing is incredible. If you swim in the wrong water in sub-Saharan Africa, this parasite will get through your unbroken skin in about 7 seconds, get into your bloodstream, start laying eggs in millions of eggs in your internal organs, make you very sick. Doesn't kill you, so it doesn't get the press that some other diseases do. But the burden on you is terrible. And if you're a kid that gets this, then you don't grow up properly, you don't go to school. So the impact on society is very great. And the numbers are truly incredible. 400 million people have this disease, and we're only treating 100 million people at the moment. But the drug is really great. So this is praziquantel, was discovered in the 70s, and is used to treat this disease. So you take this pill, big horse-sized pill, a couple of times, and you're cleared of this thing, pretty much. And it has that structure, simple structure, right? Synthetic thing, it's not from nature. The problem is, of course, if you live in the endemic countries, you'll get it again, but not quite as bad. But this parasite completely evades your immune system. It's extraordinary. You take this thing and you're cleared, which is great. And now this compound is, of course, generic, so it's very cheap. Because of market forces, the prices come down. And it's being given on a large scale by philanthropic agencies, like the Schistosomiasis Control Initiative, which is funded by Gates. So there is one problem with this drug, which is that it tastes terrible. and this is a compliance issue. If you're in an affected community and you're given this really big-sized pill, and it's the bitterest thing you've ever tasted in your life, that affects compliance. And so if you don't have a full treatment of a community, you get the acceleration of development of resistance, because partially resistant parasites will accumulate. So you have to have everyone taking this thing in areas where treatment happens. And so the bitterness is a problem. So the World Health Organization came to us and said, can you make this pill, the same, but just without the bitter taste. They tried formulating with strawberry essence and stuff like that. It doesn't really work. Chem 101, this molecule is chiral, so it has a non-superimposable mirror image. I don't know, I can't quite assess the audience here. So the chiral molecule means that it's, yeah, right. So it's like your hands, right? Hand, both the hands, but they're not the same because you can't superimpose them. You can't put a right hand into a left-handed glove, okay? So they're both hands, but they're not quite the same. They're mirror images. The molecule is like that. The two mirror images are shown. The H coming forwards and back is the only difference. It turns out that the enantiomer, or mirror image, that is effective is this R enantiomer, the H is coming towards you. The other one is inactive, has some side effects, and is the one that's responsible for the bitter taste. Isn't that great? So if you could just get rid of this, you would halve the size of the pill. It would be twice as active per weight, and you wouldn't have the taste. So that sounded great to us. It sounded like a great way that we could make an impact from using organic chemistry, which is my discipline, to affect a public health problem, to make this pill more effective. The issue, of course, is that if you start to do some research on something, you increase the price, and we can't do that. So if I had a coffee with any chemist in this audience, and I said, well, how would you do this? How would you get just the one enantiomer without the other one? Then we'd sketch out 100 different ways of doing that. Paper chemistry always works, as the phrase goes in chemistry. So you can sketch whatever you want, it'll look fine. But you can't do that without increasing the price. And that's no good. We need the price to stay the same. And that's tricky. So it's one of these irritating problems where you can put time into the solution of the problem, and the cost will go down on this scale. But there's a threshold where I can't justify putting students on a project where I'm incrementally reducing the price of something. And industry won't touch this because the net present value of this compound is massively negative. Net present value is the amount of money you'll make back from a drug if you develop it. So it's massively negative. So there's no incentive for anyone to research this in an industrial setting. So it's a public health problem which can't necessarily be addressed by academia or industry. And I thought, this is fascinating. Why is that? Why can't we achieve this through regular means? You can't take the original chemistry that's used to make this molecule and make it produce just the one enantiomer you want. That's my field, asymmetric catalysis. Can't do that. Hydrogen cyanide, you can't use in an asymmetric reaction at the moment. And you can't take the generic approach, which is this one, which uses concentrated sulfuric acid, and expect to get one enantiomer out at the end. So the existing means of making the molecule can't be tweaked to produce the one enantiomer. Okay, so that was the problem that we had. And this was back in 2005, when we emigrated from London to Sydney. And my wife and I were on a honeymoon. We did a one-way honeymoon. It was very romantic. We left England. But waved goodbye, a one-way ticket, very exciting. And on the way there in South Africa, we stopped because my wife's from South Africa. And I was thinking about this problem when I shouldn't have been. But I was thinking about this problem. And quietly to myself. And it occurred to me, well, the things that were going on around that time, around 2005, if you think back, were some pretty amazing things. So Wikipedia was blooming, and Firefox was just getting started. Linux was an established product. And I thought, these things are amazing. These things are emerging from a community effort where anybody can participate. There's no structure. And things emerge that are robust. No patents, obviously. And I thought, this is very inspiring. Why can't we do that in science? And I thought about that, and I thought, well, that's actually why I got into science in the first place, was to work like this, where you didn't matter who participated. It was a meritocracy. And something emerged at the end of it which was useful. So this is open source. The idea of open source is that, if you don't know, that it's a project where anybody can participate, all the code is freely available, and your contributions can go back into the original pot, as it were. You can make changes to the stem of the project at any point. That is not the same as open access in academic literature, which is all the rage now. That just means you can read papers. It's not the same thing. And it's not the same as open innovation, where there's a lot of secrecy. Open source is where everything is freely laid out. Okay. So the idea then was to have a project where, instead of the linear approach of doing some research and publishing it and getting a grant, and then the linear process where these hours are months or years of effort, the idea would be a very simple one, to have everyone work at the same time, using the internet, which was invented not so long ago. So that was a promising idea. I thought it was a good idea. And since then, there have been other projects that have emerged that are like this. So these are some examples. There are now more, of course. But Foldit was an example of crowdsourcing, where people could take part in minimizing protein structures using a computer game. Immensely valuable and powerful project. Other things have emerged. The Polymath blog by the Fields medalist, the mathematician Tim Gowers at Cambridge University, in 2007 and to 2008, solved a problem in pure mathematics using a blog. Again, it didn't matter who took part. You came along, made your contribution, and then waited for a response. And there are others. Galaxy Zero is a crowdsourcing project. Open Source Drug Discovery is an Indian project, which I might talk about later, which the name is currently slightly inaccurate, but we can talk about that later. So the idea was to have a scientific project, not based on code or software, but a scientific project which involved labs, where we didn't care who took part, and anybody could participate. There would be no patents, no secrecy. So first thing you've got to do is share data. That's the first step. It's not sufficient by any means, but it's the first step. So I took part in a project with some guys at something called the Tropical Disease Initiative, where there was a repurposing project. So you predict drugs for one disease that bind proteins associated with another disease and validate that slightly in the lab. And all the data got deposited on this website. And so that got published because there's a lot of data that people could then reuse. And that was an early example of this kind of project. But it's just data. It's not actually a collaboration happening where anybody can take part. And similarly with this thing, in 2010, which I'll come back to, Glaxo, in this amazing paper, which I will mention again, deposited a huge amount of anti-malarial activity data in the public domain. And in the press, of course, it's described as open source, but it's actually not the case. It's just data. If it was open source, there would be a project associated with the data, which people would be participating in. Okay. So in 2005, 2006, we started a website called the synapticleap, posted this problem in anti-oscillative synthesis of praziquantel, and sort of sat back and hoped people would help us out. And of course, no one's going to do that. And anyone in software knows no one's going to do that. There's no incentive, really, to take part. There's always the possibility that I and my team are jokers, and it would be bad for people to be associated with us. So you have to build a reputation somehow. There was a dribble of advice from people, which was great, and we've actually, this was a colleague in Queensland, suggested something, which is good, but it was a dribble of interest. So what we had to do was get funded, a core of this, and reading around the open source lit at the time, I realized this is quite common. The big open source projects usually have a funded kernel to which people respond. So we got a grant from the Australian government and TDR, which is the Tropical Disease Wing of WHO, the World Health Organization. And we started in the lab, right? So we had an electronic lab notebook. Instead of writing on paper, you put the lab book on the internet every day, and there's a new experiment. It's like live TV, If you drop something, you have to say, well, I dropped this, sorry. You have to put everything there so that people can see that they're up to date with what you're doing. And that was good, and that began this dribble of interest, a building dribble of interest from the pharmaceutical industry, the process chemistry industry in particular, who said, no, you're going about this all wrong. Your rarefied academic approach, which you got funded, is not the way to go here. This is this reaction we were trying to do, which is still impossible. You shouldn't be doing that. You're never going to work. It's never going to work. You're never going to get the price down. You should be doing a more seasoned, old-school approach called a resolution. Any chemist here will know about a resolution. It's what Louis Pasteur did when he picked out tartaric acid crystals from things. You can pick out different enantiomers of things if you make diastereomeric salts. So you can pick out salts of things if you crystallize it in the right way. So we changed the direction of the project 90 degrees because of the input from people. And that's the point. We didn't make a strategic mistake of doing this for 2 years and then publishing a failure. People could come in from day one and say, no, don't do that. That's a stupid idea. And so we hit a roadblock. We realized that you could take praziquantel and chop a bit off, revealing this thing called an amine, and you throw in something called an acid, and you get a salt crashes out. And you can, from that, you can get one of these mirror images out rather than the other one. It's called a classical resolution. And we couldn't do this in our lab because we didn't have an assay for assessing whether this is working, and we didn't know where to start. If you've ever done any resolution in chemistry, it's this black art. It's incredible. It's magic. People just know how to do it. You have green-fingered people who can just make crystals crash out like that. So we had to admit on LinkedIn, of all places, that we didn't know how to do this. And thankfully on LinkedIn, there's a room of 1,000 guys who are process chemists who read this, and 5 companies came forward and said, just send us some of this thing. Just send us some of this thing. And we'll screen our acids, and we'll do the work, and we'll give you the data for free. So we did send a gram to these guys in the Netherlands, and within about 2 weeks, they'd come back, A, with an assay method, and B, with a hit of a chiral acid that we could put in this thing to get this thing crashing out. So we then took that, and over about 6 months, we optimized it until we got to our final solution, which is shown here. So again, this is a bit of chemistry, but it's very simple. So you take the stuff you get anyway from the pill, you chop a bit off, you resolve it, and you put that bit back on, and you get one enantiomer out, so about 33% yield or something, about half of it back. Enantiomer pure, beautiful crystals of this perfect enantiomer pure material. Right, so that worked very nicely, and the key about that was the input from the chemical industry, and the key about that was that we didn't know the people who helped us before we started. So because it was open, the best people came to the problem and helped us accelerate the research. It was just that 2010 was the year between January and about November. So exciting to see this and these inputs coming from people that helped accelerate the research. Inputs that were freely provided, right, for whatever reason, PR, demonstration of core competence, the irrepressible nerd in all of us, right, that you see something being done badly, you just can't, you can't help step in. No, 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 no. You just want to do this. So if you fully explain how bad you are, people will help you out. Okay, interesting thing happened was that at the same time we were doing this, we were getting a little bit of press on chemical blogs, various chemical blogs. And there was one that's widely read in chemistry called In the Pipeline, and we were featured there as an approach to an unusual approach to a problem. And half the comments there were supportive, and half were visceral in their ridicule of what we were doing. Presumably because we were trying to open source a profession. We were trying to say that we could do process chemistry as a bunch of amateurs. And one comment said, don't worry about these guys. The World Health Organization has contracted a company to solve this now. And I read that and I thought, well, that's not right because we're paid by the World Health Organization. And so I wrote to our contact in the World Health Organization and said, well, this is just rumor, isn't it? And they said, no, we've been paying a company to look at the same problem in secret in competition with you. So I didn't know that, but it turns out that they'd been working on it for a few months full time, being paid, to do that. And they came up with this solution down here, which is very similar. You can buy this on a ton scale. You can't buy it in a kilo. You buy it on a ton scale in China. And we didn't know that because it's not a name of the catalogs. But they took this and did the same kind of resolution idea and then put the thing together and got the same product out. Now, I don't know which, if you cost it, I don't know which is more efficient because I can't get the data from the company. And I don't know which project is more expensive because I don't know how much work they did on the project. But it's interesting that the professional outfit and the public open source outfit came up with essentially the same idea at around the same time. So in retrospect, even though I was livid, in retrospect, it was a useful benchmark. Okay, so the perceived advantages of working in this way are, for me anyway, are transparency, so everyone can see what's going on. The public can see that you're spending funds wisely, right? So there's no hint. If this was climate change, that would be important, right? That people can see that the research you're doing is robust. The starfish is an analogy from software development. The point about a project when it's open is, of course, it doesn't matter if I fall under a bus. So the project can then restart with whomever because all of the data and ideas are recorded. So it doesn't matter if the leader is taken out of the project. Speed was a big thing for us, so the research was undoubtedly accelerated because people we didn't know who had a higher level of expertise than we did came along and contributed freely. And lastly, you need food. So you need to provide people with something. You can't just expect people to help out. And that's a problem with a lot of projects that are sort of suggested on the web, is people expect that the public have an infinite amount of time and goodwill to contribute towards other things. And they don't. You have to be quite serious about, well, in our experience, what happened when we got going, we had the inputs. You provide a kernel, and then people will respond to that. Various things came up as part of the project, which were quite interesting. When you work on the internet, you want people to find what you're doing. And it's not that easy, actually, with chemistry. So Google, if you go to Google and there's a little box there, you type in it, right, words and letters. You can't you can't draw something there. So if you want to search for a chemical structure, you have to translate the chemical structure into a text string, which is fine in chemistry, but to automate that from our lab books, an undergraduate in my lab, over the course of a weekend, wrote something that took an image from our lab books and used optical character recognition to construct a live image of the molecule, translated that into a smile string, and then asked the human if it was right, and then when you tick that box, that's inserted into the lab book entry. So now, the lab book entry is Googleable. So you can find out what molecule is being worked on using Google right now. You don't need to wait until the paper's published, you can find it. And that's great, right? If you're a chemist, you want to know if anyone is having the same problems that you're having. And so if you can use Google to do that, that's a more efficient way of than waiting for a paper to be published. We're applying, I'm going to talk about another story now, but we're applying it, the same idea to catalysis, which is my first love. making molecules in a catalytic way, preferably as single enantiomers. And I have an undergraduate who's trying to do this at the moment with this lovely open source catalysis project. The undergraduate's really going to town on capturing the beautiful kind of, what's the word, sensory pleasure of being in a lab, that, apart from the smells, which we can't do on the internet yet. So capturing the images of little bits of crystals forming, or the fact that a reaction changed color a little bit, or something that went wrong, images, plus raw data, plus thoughts. She's really maintaining a fantastic lab book. And it's getting a lot of responses as a result, because it's a very attractive thing to read. Anyway, she's trying to do that. The idea behind that is, if that works, we're going to try to attack a big problem in catalysis in the future, which is about predicting catalysts for chemical reactions, but that's a big project for the future. Okay, so so for a project like this, you nearly have everything. You've got a lab book here. You've got a place where you can talk about what you're doing. You've got a wiki, which we use for maintaining a kind of project status, where we're up to. You've got places where you can publish work like this. So this was in PLOS NTD. So places, journals which will take research which has already been in the public domain, such as PLOS. I'm pretty sure Nature and Science are fine with that. Biomed Central are fine with that. Pretty sure PNAS are fine with that. So increasing number of publishers will take papers that have already been in the public domain, or data which has been in the public domain. The biggest chemical publisher, the American Chemical Society, won't touch it, but that's okay. And then you also have other people talking about the research you're doing on other blogs. So you nearly have everything for a very interesting cycle here, but there's something missing, which I'll get to later, something crucial. All right, so the question I get then about this is, of course, the project a known drug was involved in that project, something called praziquantel, a drug which was off patent. Very little IP associated with that. So the question is, can you do drug discovery in this mode, where you are trying to find new molecules, where there is associated IP and patents and so on. And the idea then would be to take something which was known, some public domain hit, and again, rather than being linear, you work together at the same time to produce an improved molecule. So it's called a hit-to-lead campaign in drug discovery, which is quite intensive on the chemistry. You have to make molecules and test them. So, I mean, methodologically, the answer is yes. You have the same thing. You have a lab book, places where you discuss things, ways to bring light social media things to bring people in. Wherever people are, you go there and talk about the project. So Twitter's quite useful for that. The question is, can this work then if it's in a drug discovery project where people may be nervous about IP? And we were asked that a lot, and we were talking about it so much that we got tired of talking about it, and we decided to try and do it. So I got money from the Medicines for Malaria venture, which is part funded by Gates and the UK government, to try this kind of idea in malaria drug discovery. Now, malaria is interesting because the net present value of compounds for malaria is about zero, roughly zero. So from neglected tropical disease up to malaria, which is not technically neglected, it's slightly more financially interesting because you're not going to make any money really, but it's a bit closer to the sort of break-even point of whether the disease is profitable. So the starting point is this truly amazing paper from GlaxoSmithKline that came out in 2010, Nature paper. And they did what is known as a phenotypic screen. So many of you, if you haven't been in drug discovery in the last 20 years, you'll think of drug discovery as being a rational process where you have a target and you guide some molecule in to fit into that target, and you generate some biological activity by doing that. The emergence of high-throughput screens using organisms, in this case the parasites, living inside red blood cells, has meant that you don't need to do that anymore. You forego knowledge of what the compound is doing, and you gain potent compounds that work with a cell, the phenotypic screen. So Glaxo did a phenotypic screen for malaria, red blood cell, parasite inside, of their entire library of, I don't know, 2 million compounds or whatever it was, and published the best hits. So the ones that were non-toxic to mammalian cells but potent in this assay. And they published 13,000 starting points. Changes the landscape completely of malaria drug discovery. Because now there are so many hits of molecules that are non-toxic and potent that you don't need any more for the moment. We're kind of done. What you've got to do is explore these. Really extraordinary paper, really extraordinary. I met the lead author and asked him why he did it. And he said he wanted to increase the interactions between GSK and academic groups. Simple as that. And that's happened, certainly. because lots of people are now working on these compounds. We picked a few, started with these ones. So this is a pretty simple molecule, very nicely potent, very nice medchem properties that mean that you can change the structure and still be in the kind of sweet spot for medchem. And there are some backups. So we started with these. And what you've got to do, of course, is just make some molecules. You need principles for how this project works. So based on the stuff with praziquantel, we formulated 6 laws of how this thing was going to work right from day one. So as soon as we got funded, everything's open. Anyone can take part, no patents, no flame wars, right? So it's best to suggest things rather than criticize. Don't send email. And the project is a project to which people are contributing. My lab's funded to do it, so we're leading it, but the project will last after we have gone. Wikipedia is not called Jimmy Wales's encyclopedia. That would be a disaster, because no one will contribute to that. It has to be something which is not owned too heavily, I think. So you make molecules, and so some medchem, just for those of you who are thirsty for some structures. This compound and this compound were the 2 hits we went for. We confirmed that they were extremely potent, very attractive. There's an ester there in the middle. Anyone who does drug discovery will think, well, that's going to get cleaved when you put it into an organism. It's just going to get sliced. So you investigate whether these are prodrugs, and you synthesize halves of these molecules. Those are all inactive, so the prodrug thing was a no-go. You may make variations. This, to anyone who's an organic chemist and has ever done any peptide coupling, is a joke. So feel free to laugh at that if you're an organic chemist. It's funny if you know why. This turned out to be in a really great compound. So the potency went up by an order of magnitude when we got to this compound. Unfortunately, this is pretty insoluble, so you start to get problems with solubility. That's not good, but it was very potent, so we went after this. And so all of the data is being deposited on the lab books, and the biological evaluation, as soon as we receive it, is posted to the lab book. So you do other stuff. You try and take the ester out by making it amide, so you do an oxygen to NH replacement. It's the most amazing thing, medchem. You make this tiny little change, and you obliterate activity. So changing an oxygen for a nitrogen just completely inactivates the compound. And that made us think we were in trouble here, because that implied we couldn't really do anything with that ester. So the other really potent compounds, though, we made a bunch of analogs over here and got down to picomolar activity. But it's a little bit like brick dust, so it's very, very difficult to dissolve. So highly potent, which was great, and we were there in about 3 or 4 months, but unfortunately very, very insoluble. And so we started doing some in vivo work, and it turns out that the molecules, well, down here, put it into a mouse. It turns out that the lead compounds from Glaxo, that ester is a problem. The molecule goes in, enters the blood, is fine, but it gets cleaved in about 4 hours, and that's about 4 or 5 times quicker than you need for malaria. You need it to be there for about a day. So that was a problem, and we needed to get rid of that ester. That was the idea. We found other things that were good. It passed the Herg assay. This gametocyte thing is very important. So most antimalarial drugs work by the same mechanism. There are very, very few that work on a certain stage of the parasite life cycle when the mosquito bites you and the parasite goes into the mosquito from your blood. That's a crucial stage, right? Because that's when the disease is spread, essentially. So if you can block that stage, kill that stage of the parasite, then the disease is then inhibited from spreading, which is great. So that's called a late-stage gametocyte assay. And these new compounds we found, the potent ones, were very potent in that assay. So that's actually very exciting. That's quite unusual. But given the solubility problems, we didn't pursue that. We pursued the other series, so this Elpuro series. So it was all about public consultation. So we had a big public consultation, and we had a commercial top 10 list. These are all commercially available, amazingly, usually from Eastern Europe. There are lots of labs that have compounds in, and you can buy 10 milligrams from a company, and they'll ship them to you wherever. So we formulated a list of the most relevant, top 10 commercial compounds, and bought those. And then we had a top 10 list of synthetic, and we assigned ourselves some of these and assigned other people other ones, and we kind of got to work on completing the set. I made a bunch of these things, so variations. Again, you can see the red changes. It's all about getting rid of that ester, right? And sadly, the news wasn't great. Basically, everything failed. So this ester was really needed, and we couldn't change it for anything else. This is the original list. This is an example of, we were assigning people to make various compounds. We have every 2 months we have a public meeting where it's streamed online and it's recorded and dumped on YouTube, so people can see what we're going to be doing in the next 2 months. It's an important part of the process, so people feel as if they're up to date. the contributions we got were very good. So there was some nice dialogue. You don't need to read this. This is, there's a little motif in one of these compounds, which is like a red flag to a med chemist. It's there, and you think, actually, that's going to be a problem. That's something called a PAINS, or a pan-assay interference compound or something. So it's a thing which always interferes with high-throughput assays. And my student, somebody called Jonathan Bale, who's at Monash in Australia, invented this term and noticed that there were these problem compounds. He wrote in on our blog and said, by the way, you should be aware, your best compounds have got this problematic thing in. You might want to be careful, because it might be a false positive. And so my undergraduate, who just finished her first year undergrad, wrote and said, actually, if you go back to the original paper, they've filtered out those compounds. So we don't need to worry. And he wrote back saying, well answered. I just loved watching that, because Jonathan and Zoe didn't know who each other were. And Jonathan's a professor and Zoe's an undergrad. And they were having a constructive dialogue online without me being involved. This happened overnight. So I wasn't involved. And I thought, this is what it should be about, right? It's a level playing field where anybody can contribute to a constructive dialogue. We had another go at the last 3 compounds we couldn't make. Really annoying ether compound that we still can't make. This thing called the sulfonamide, and this thing which we ditched eventually. But this sulfonamide here, we were trying to make it in the lab and we couldn't. And so we made this little poster up to say, these are the wanted compounds, these 3, like dead or alive, right? You've got to try and make these things, because they're obvious replacements for the ester. And this amazing Twitter thing started up. So there was a postdoc. So we couldn't make this thing. We were trying to make it and we couldn't. And this postdoc in the University of Edinburgh wrote in and said, hey, I've got my thesis out the way. I could do something. I said, well, how about making a molecule? And he said, okay. And he started in his lab in Edinburgh, and he started posting the data on our own lab books. And he was posting spectra so we could see what was going on. And he'd misassigned this, so I was able to sort of say, are you sure this is right? And we had a good discussion about what was going on. And then he eventually made it. And he said, good morning, Australia, have a sulfonamide. He made it. And then he packaged it up, and he put a picture of himself with the Jiffy bag with it in. And it was so great. So he sent that off for testing locally in Dundee. It doesn't matter where you test things, because you can include controls. And it was great, except it's inactive. So very unfortunate. So the compound is inactive, so we can't change it. So we, at the moment, have ditched the series. We're about to publish this work. And we've ditched the series and gone to another, which is looking really great. This compound is extremely potent. Our first round, again, we changed little tiny things about the molecule. Everything was inactive. But we're now on round 2, where we have smaller changes that we think should make a more constructive difference. Let me just say a couple of things about the way this works. Data management, very straightforward. Software people have solved everything in terms of managing projects like this. Data management using GitHub is very straightforward. This is a file with all of our compounds in, updated continuously. And every now and again, we deposit to Chemble, which is the free online database of biological activity of chemical compounds. You get a DOI with this, so you can cite it. Project management, we had a real issue with until we realized that actually GitHub was a great solution for this too. The software guys are way ahead of us. Really useful project management tool for supplying a to-do list. And you can label things and assign people and close things. It's a tremendous resource that's just sitting there, waiting for the communal management of a scientific project. So that's been very useful. And you can browse, the current issue list at that address, and you can just see what we're currently working on and who's contributing to what. It's been a very good experience. So, nearly got everything. The thing we don't have is data from everybody else in the world. So we're the only ones doing this, and everyone else is publishing. Everyone else is perhaps keeping back negative data. We're putting the data out there, but what we want, of course, is to have the data on every chemical reaction that's ever been performed in the history of the world for us to play with. You need to play with data. Like I said once to Larry before, you need the data to be able to play so you can release your inner child and play around with this stuff and just start moving things around and spotting trends and elucidating things without worrying about whether something's behind a paywall. So that's unfortunate. Chemistry has never had its human genome moment where everything is put in the public domain. So we're way behind you biology guys on that. Okay, just 2 more things. The elephant in the room, patents. Can you develop a drug without a patent? On the face of it, the answer is no, because that's what we all think. In fact, of course, the answer is yes, because I think I'm right in saying that penicillin and the polio vaccine were brought to market with no patents. Intellectually, the question is an interesting one. We avoid patents because we don't want barriers to the collaborative process. We want people to be able to participate. But there's some people writing interesting things on this issue. Bolgen and Levine, two academics in the States, they've been examining whether patents help innovation or hinder it. Their ultimate philosophy in an article in a book is that patents shelter an organization from the need to innovate. Quite controversial opinion. They use a very nice historical example of the rise of the German chemical industry in the late 19th century. In Germany, you couldn't patent a molecule. You could only patent a process. In the UK and France, you could patent a molecule as well as a process. And we all know what happened between 1890 and 1930. Germany just went, it dominated the market because the thesis is that it was forced to continually innovate. Another paper that was published more recently, this study by someone at MIT, that examined the research work arising from from genes that were patented by Celera versus those that were not, that were part of the Human Genome Project that were never patented, found that there was more research activity on genes that were not patented. I don't know if I agree with this, but it's interesting data. Okay, last thing I want to say is an analogy. Imagine you're watching the Olympic sprint. It's exciting. It's exciting because the gun goes off and people run, and you watch the process. I enjoy watching that. It's exciting. If you put tunnels there between the gun and the starting line, and you just wait for 10 seconds, and someone pops out at the end, that's a dull event right there. There is excitement in seeing the process. So there's excitement for students, for the young people, to see the process of science. For us, that's not the issue, though, is it? For us, we are the participants, so there's a different analogy. Imagine you're the runner, and you're going to run the 100-meter sprint, and you're in a tunnel. That's boring, right? That's boring race. You're going to run your heart out, and you're going to pop out at the end, and have I won it? I don't know. It's boring, because you can't see what everyone else is doing. Much better and more exciting is if you're a contestant there. I mean, just looking at this gets my heart racing. You're there and you think, okay, I'm going to race against other people. If people are pulling ahead of me, I've got to change what I'm doing to be faster. The thing about open source is it's competition in an open arena. It's this bizarre paradox of openness and competition, cooperation and competition at the same time. That's why it's so exciting. It's a true competition. You're not sheltered by anything. It's actually quite brutal. Just some people on the project in pictorial form, a few people from around the world, and some names of people who've taken part. It's quite a difficult slide, this for an open project, because there's like anonymous at the end here, because people can contribute without telling you who they are. Funding agencies, particularly WHO and MMV, for funding. And thank you very much for your attention.
Hi, Matt. Enjoyed your presentation. Practical question. Your praziquantel enantiomer-specific synthesis,
yep.
Did it solve the problem, or are you hampered by in vivo racemization afterwards that essentially makes the process nice, but the solution is minimal?
No, it solves the problem. It doesn't racemize. The problem is that the solution was delivered, and we know that there are companies and other academic labs that are making use of it because it's open, but we don't, the process of taking that up to a process scale by a factory is now, unfortunately, behind closed doors. So I'm not in touch with that anymore. It's been taken on by WHO. Consultation is happening. We don't know what the outcome's going to be. But the solution that as delivered is a viable possibility.
When I heard you talk a year ago, when I was so excited, you made comments, but didn't this time, that I wish you would say something about, because as I recall, you were shocked and gratified by the responses to you from industry and less excited about the responses from academia.
Yeah.
Would you say something more than that sentence?
It was extraordinary. So 75% of the inputs on the praziquantel project were from industry, 25% from academia. Exactly the opposite of what I was expecting. To my mind, industry, and we've gained more interactions with industry on the Malaria project, where again, the GSK guys, to be honest, are the most willing to share. The GlaxoSmithKline guys, are the most willing to share. They get it. It's a little bit skewed because the GlaxoSmithKline lab in Tres Cantos in Madrid in Spain is a not-for-profit arm of GSK, right? So it's a bit different, but I mean, I gave this talk in London in September, and at the end of it, a guy walked past and said, "Yeah, cool," and he was in charge of drug discovery for Novartis, right? The academics didn't say that. I think in academia we are becoming incredibly secretive, and I just don't get it. I just don't get it. Yeah, well, I mean, that's just very unfortunate that's the incentive. Yeah, so, I mean, it's difficult. So promotions committees don't know what to do with this. Definitely, and it helps to have DOIs, but it's not the same. It's not the same as a paper in a traditional journal, so it's difficult to explain. I would say most of my colleagues think that we're doing open access. We're trying to publish our work in open access journals. So completely misreading the idea that people take part. But academia, I think it's just become skewed, and I don't know. I mean, you can be an idealist about this, but when I was a kid watching science on TV, it was usually American professors, actually, who were talking about space and dinosaurs or whatever. I wanted to be a scientist because I just thought you'd do work and you'd be interested in what you're doing, and you didn't have to worry about politics. And then you get in the job and you realize, Jesus, it's politics and everyone's competing with each other, and it's crazy because we're not being efficient. So yeah, it's exactly the other way of what I thought it would be.
Thank you, Professor Todd. Getting back to the sharing of information and tying into the Germans and the Swiss, I know that the Germans developed IV ozone to treat certain health conditions such as cancer and whatnot. I found for myself doing it the IV way is you risk infection and drying out your tissues, et cetera. But the rectal
ozone, did you say?
Yeah, but rectal insufflation now you can Google it, PubMed, Medline, and rectal insufflation is accepted means of delivering higher forms of oxygen to. It's been for Dana Swenson that would have been ideal to treat her condition, but it's kind of still considered an alternative modality because it's not. It's kind of what you're trying to establish, treating the condition, not trying to make a whole lot of money and getting the patents on it, et cetera. So I was just kind of curious, for malaria, for instance, has there been any study or research as far as trying to treat that condition with more of gases or things of that nature? I know it sounds kind of radical, but a gas will get to every, crosses the blood-brain barrier into the bone marrow. There's no place that a gas can't get. And mightn't a gas be also a means of delivering certain pharmaceuticals? So if that jives with you at all, thank you very much.
I mean, the answer is no. I get people writing to me about things like nascent iodine. And you mentioned a higher form of oxygen. I don't know what that is. Sounds like a religious form of oxygen. Okay. No, so I'm a big believer in the value of chemotherapy. Chris Walsh has had the same question before. I think that these molecules actually kill the parasite. Using something like gas, you can do it if you want. Get some data, put it in the public domain. Let's get it peer-reviewed. But these things are working just fine.
How do you think this process will work that you've developed with the antibiotic issue that we discussed earlier today that costs $1 billion to get something through the FDA?
No, it's an interesting question. So the patent thing is completely dependent on this idea of net present value. So we started with neglected tropical diseases because it's obvious that this is an approach that might work because there's not a profit to be made. Malaria is more even. If we then apply it to something else, which we'd like to apply it to, say, TB, there is a potentially positive net present value of one of those compounds. So it becomes more difficult. We're trying to do that. What I'd love to do is start something like this in dementia, where the net present values seem to be colossally positive. I don't see, this is a large philosophical conversation there, I don't see why you can't apply this model to any disease. I don't know if it would work, but it can be applied. You will always find people who are willing to participate, willing to take part in a project like this, don't have a conflicting interest, they don't have a contract which prevents them from doing that. The antibiotic thing is an interesting question. Chris laid out the awfulness of the development of resistance. The same thing's going to happen with malaria. I mean, we have the combination therapies for malaria with artemisinin in it and other things, and there is now resistance to those things on the Burmese-Thailand border, I think it is. It's inevitable. It's inevitable. So you have to have this constant flow-through of new compounds. We have a healthy pipeline for malaria, but we don't have the compounds that are validated, that have finished phase 3. So you need other approaches. We need more compounds. We need other approaches. This is not trying to invalidate a commercial approach. It's trying to just do something different to shake it up a little bit. So it could technically be applied to anything, I think.
Very good. Thank you very much. I'm really sorry we have to go on.